Yeah, 1 and 3, I put them together because it made sense to me. I don't know, I feel like 3 is kind of introductory also. But also 2, I guess, is chemistry. You guys are supposed to know that crap too, but yeah.
I'm still reading chapter 2 for some odd reason.
It's like 20 years since I had chemistry.
All right, so let's try to go through. I hope that it doesn't take too long because we've got 4 chapters to do these questions with. But if we have to start picking and choosing the questions instead of going through all of them, then we'll just do that because I don't want to be stuck here forever. So we'll see how it goes. All right, so chapter 1, we're doing our intro to microbiology. Basically getting introduced to what idea of what a microorganism is and what it is to be alive, and definitions of the types of organisms that we are dealing with, like prokaryotes that don't have a true nucleus and eukaryotes that do have a true nucleus. So what are microorganisms? Microorganisms are organisms that can't be seen with the naked eye, so we have to magnify them somehow. Like that's a pretty easy one. So what aspects of microorganisms do microbiologists study? We might look at like the cell size, the cell shape, the physiology of the cells. Do they make any toxins or not? How do they interact with any potential hosts that they could be parasitic to? All of this sort of stuff, and how they interact with the host is also incredibly important with regards to any immune responses hosts might be having to microorganisms. So the cell theory of living things has to deal with the idea that all cells come from other living cells. They had to come from a living cell that was able to divide and make that cell. They have to have DNA. They have to have at least the cytoplasmic membrane to contain what's in the cell. So that's the idea of the cell theory, and that's why we say that viruses are not alive, according to that theory, because they are not made of cells. They do have a genome, but their genome isn't even always DNA. For viruses, which we will learn in chapter 6, we haven't gotten to that yet, but chapter 6 will tell us that viruses can have RNA or DNA. They can be single-stranded even for their genome. So they're just kind of on their own. They can't divide without living in a host cell. We don't consider them to be alive, technically, according to that. All right, a pathogen is, so what is pathogen, what is disease, and are they always related? So no is the answer to that last part, right? And this is why. Disease isn't always caused by microorganisms, and infections, right? So you can have cancer. That was a really good example. Cardiovascular disease, another one, that these are not associated with having an infection necessarily. So a pathogen is anytime you have a microbe that is causing disease in another organism, and disease, that's any deviation from the normal state of that organism. A negative deviation from the normal state. A prokaryote, what does that mean? And eukaryote. And which ones came first? As far as evolutionary, biology goes, I don't find it particularly interesting, but it's something I have to teach you guys. A prokaryote, pro means before, karyo is dealing with the nucleus. If you've ever heard of karyotyping, it's like getting the chromosomes all lined up and counting them. Like you've seen those pictures of the chromosomes, right? With the X's near one another. That is dealing with the nucleus. And so prokaryote means before nucleuses existed. They don't have a true nucleus. Their genetic information is just kind of floating in an area in the cytoplasm called the nucleoid region. So whatever, I guess, I don't even know what class that was in. Sorry. I guess there was that girl that like randomly left the class, I guess at some point. I don't know if you guys remember that, but there was at some point. But apparently she had an emergency. But I didn't know if you guys knew who it was, but I was just gonna let you know that was what's up, but nobody knows what I'm talking about. So that's okay too. That's okay too. All right. So that's before the nucleus. And then we have the eukaryote. Eu means true, karyote again, nucleus. So the eukaryotes have a true nucleus that are membrane bound. They also have true membrane bound organelles like mitochondria, endoplasmic reticulum, Golgi apparatus, all chloroplasts, all of those guys. Evolutionarily speaking, it makes sense that the prokaryotes would come first. And it also kind of makes sense whenever you look at, when we get into talking about, I guess more in chapter 4, but the endosymbiotic theory that, some of these early cells came together and now have become organelles within what is now true eukaryote, which is really, I think, kind of interesting. Turns out that mitochondria, our energy powerhouses, have their own genomes that are circular, just like bacteria have. They have their own ribosomes, which are the same as bacterial ribosomes, not like our ribosomes. And they divide like bacteria do as well. There's some other aspects that associate with bacteria rather than us. So indicates that at some point, maybe those guys were bacteria. I think that's fascinating. I am not interested enough to study it though. So the scientific method, we know, because I say it quite a lot in a lot of the lectures and in the reviews and everything, but this is done through deductive reasoning, where we might make an observation of our environment and then come up with a hypothesis to explain what's going on, and then perform experiments in order to answer, is this in fact what is going on or not? So proving or disproving that hypothesis based on those kind of trials of experiments. Microbes, do they have any other functions other than causing disease? Of course they do. They break down, dead and decaying organisms. So we don't just have dead things just lying around. They actually work within our bodies to help break down food and provide nutrients to our system when otherwise we might not be able to do so for ourselves. They actually provide protection for us as well. I always explain it to people as being like taking up the stadium seats of all of our normal bacteria. Our normal microbiome is taking up our stadium seats in our body, in our gut, on our skin, whatever it is. Then the bad guys can't come down and have a seat. So, and they can help fight off those bad guys too. That's why it becomes a problem whenever we have antibiotic treatment that kills off bacteria in someone's gut, and Clostridium difficile, C. diff, can overgrow and cause, pseudomembranous colitis as a result of that. Pretty nasty stuff, but yeah. All right, so what was next? Oh, right, and food. Obviously, we can make use of these microorganisms for making things like beer or wine or cheese or, kimchi or pickles or whatever you want. Fermentation is really important in that, and that microbes like bacteria and yeast are able to do that for us. Okay, taxonomy, man, talk about boring. Okay, what are taxonomy and binomial nomenclature, and what is the correct way to write an organism's scientific name? Taxonomy has to deal with the way in which we are classifying organisms based on phenotypic traits, which is like observable traits, or even their genetic makeup, which we can look at that now and compare organisms that way as well. And based on all of these traits together, grouping them into certain groups and levels of classification. The largest one overall is, of course, going to be the domain category, and that is going to be including eukarya, bacteria, and archaea. Those are the 3 domains. Yeah, and then we have basically after that organization, kingdom, phylum, class, order, family, genus, species. I have to say it all at once or it doesn't come out in the right order, but it just feels right if I say it the right way. So, yeah, if we write it out, kingdom, phylum, class, order, family, genus, species, KPCOFGS. I don't have a little acronym or memory thing for these, but if you guys, I would recommend Googling one and trying to find one that helps you remember that whole situation. There used to be one that was like King Philip.
Yeah, did Philip come over to eat for good spaghetti?
Yeah. That works. So yes, anything like that that helps you guys remember that order, because there will be questions asking. It might not ask you to list like all of them in order, but it might have like 3 of them and ask you, which of these has the 3 in the correct order. And then we have binomial nomenclature. That is like how we call, humans are Homo sapiens, or we have E. coli. That's an abbreviation for Escherichia coli, Staphylococcus aureus. Those are examples, of course. So binomial nomenclature, that's the 2 names. The first name is always going to be capitalized. The second part of the name is going to be lowercase. So in Homo sapiens, Homo, the H would be capitalized, and then in sapiens, the first S would be lowercase. Well, all the S's would be lowercase, but and then if you're typing it on a computer or it's being viewed electronically, then you want to be italicized. Otherwise, if you're writing it by hand, you're supposed to underline it. So that's how that's supposed to work. And then the levels of taxonomic classification, we just did that. So cool. All right, so that was chapter 1. So I had next up chapter 3, which I felt was like, again, a little bit more of an introduction into the concepts behind microbiology and how microbiologists work. We're talking about the 5 I's. These are basically steps that are involved in identifying an organism, a causative organism, particularly either for research, but it could also be for diagnostic purposes. For the 5 I's, we have inoculation, incubation, isolation. I can never remember that. Is it investigation or information? I think it's investigation. I don't know if that's the right word, but it's something like that. I wish I had written it down before. I feel really bad. I'm just gonna look it up. I'll tell you the wrong thing. The concept either way is the same, and that's the one I could never remember as before. I don't know why I can't ever remember that one. I can read it all day on a slide and then immediately forget it. So I apologize for that, but 5 I should have made a microbiology. Inspection, which again, to me, investigation would probably actually be a better word for it, but the official word is inspection. And then we have identification, which I actually knew that one. I just couldn't get past that 4th one. So, okay, so inoculation is going to be dealing with actually making the culture, introducing that bacterial sample or whatever swab or whatever is into media in order to grow it up. Incubation is putting it at the correct temperature with the correct gases that that organism needs for the correct amount of time to allow it to grow. Isolation, if you have a mixed culture, if I do a swab on the floor, but I'm only interested in one certain kind of bacteria on the floor, then that has, then we need to isolate that bacteria somehow. The most popular ways of dealing with that, one of them we did in the lab already, which is the streak plate. And then the other one is serial dilution, which is exactly what it sounds like. We're just going to take the cultures and dilute it out with either broth or saline, and then plate it until we get individual colonies. Then we have, so that's going to allow us to get those individual colonies. Then we have inspection, which I would have rather had been investigation, but these are essentially looking at, colony morphology, cell shape, the rate of growth, the requirements for growth, whether or not they can grow on certain kinds of media, whether they're gram-positive or gram-negative. All of these aspects and more allow us to collect this information about this organism that in the end, which is the 5th I, would be identification based on those results of everything and bringing it all together for us to be able to identify that organism, that species. So that's the 5 I's. So that does bring us into a lot of these concepts that are going to follow after this. A culture, this is a term that we use for growing up a microorganism. And no, we don't only use this for bacteria. You can culture mold, of course, but and that includes yeast. But of course, you can also culture viruses, which we'll get into the, I don't want to say the details of it, but that's, we'll talk about viruses more in chapter 6. But yeah, you can culture viruses. You can use human cells even for that sort of a thing. So that's an interesting thing to remember, but you don't only culture bacteria. The media has to be sterile because you have to be able to know that the thing that you're running the tests on, of course, is the only organism in there. Otherwise, how do you know the results from your organism or the contaminant? And that's why aseptic technique is important. Aseptic technique prevents any cross-contamination. Either like if you're working with your organism, keeping things from growing in your organism's media that might inhibit it, keeping things from growing in the media that would cause erroneous or wrong results in the tests that you're running, or even with aseptic technique, keeping whatever organism you're studying. If it's like tuberculosis, you want to be sure that aseptic technique is being going on, so you're not carrying that out of the lab with you when you're done with your research at the end of the day. So that's why that would be important for it. Agar is made from sugar, it's polysaccharide, a sugar-based molecule from algae, and it gives a solid base for media for microorganisms, for bacteria specifically. The different kinds of media, it just kind of depends on how you might approach this question. If we're talking about a general use media, that could grow up a whole lot of different kinds of organisms versus things that could be selective or differential, for example. We also have enriched media for fastidious organisms. And let's see, they have, if media is enriched, it is for fastidious organisms, and those fastidious organisms require that certain like vitamins or other things be supplied to them because they cannot make it for themselves. It has to be present in the media. That's just like us, where we need our vitamins provided to us in our foods, especially like when you think of like vitamin C or B12, stuff like that. We can't make that for ourselves. So we need that to be supplied to us in our media in a way. So that would be the enriched media for fastidious organisms. Then we have complex media. Complex media is basically media that has an ingredient that cannot be chemically defined. We also have selective media. Selective media means that it has an ingredient in there that either prohibits the growth of certain organisms or favors the growth of some organisms, but doesn't allow growth of other organisms. A great example of that is mannitol salt agar. Okay, you're going to be hearing a lot about mannitol salt agar, but it's used to grow up Staphylococcus organisms. It is salty medium, and that salt that's in there is not hospitable for a lot of microorganisms. So that is the selective aspect of that media. So only organisms that can withstand that salt will grow. The other thing about mannitol salt agar is that it is differential. Differential means that we can grow several things on that maybe, but they will have reactions to the media or products made from using the media that cause a color change. A lot of times that's based on a pH change. Most common one, of course, is going to be phenol red, and we'll become familiar with that in the lab when we get to that. But yeah, so phenol red will react to acid that is produced when you break down mannitol, the mannose sugar in the mannitol salt agar. Staphylococcus aureus can break that down, and so it creates acid byproduct and turns the media yellow from its original kind of reddish color. So that allows us to differentiate, based on that reaction that's going on. So mannitol salt agar is both selective and differential. Of course, we have transport media for transporting things. Like the other stuff, like assay media for specific tests that we're doing. Those are kind of obvious ones. We talked about growth factors already. Different types of hemolysis, right? So blood agar is technically an enriched media. It helps to grow some fastidious organisms. It's also a differential media. So if something can grow on it, we can determine if it is alpha hemolytic, beta hemolytic, or gamma hemolytic. Hemolysis is breakdown of red blood cells. So if something can completely break down the red blood cell, we'll have a clearing around the growth area where the blood cells are broken down. That is beta hemolysis. It's true hemolysis. If we have just greenish color from some breakdown of the red cells, but not complete breakdown of the red cells, that is alpha hemolysis. And then gamma hemolysis is when you see no hemolysis. I don't know why it's called hemolysis. I have no idea, but it is. That's the word for it. So colony, we know colonies come growing up from one single cell. That's just a pile of cells that have divided over and over and over again on top of each other. Now, it's crazy to think about if you plate something like we do in our lab on a plate and you see a colony that has grown up the next day on the media that came from one cell just the day before. So that's kind of cool to think about that. So that's what a colony is. We already talked about streak plates and dilution, serial dilution to get individual colonies for isolation. All right, so identifying microbes from one another, some of the tools. I mean, how I answer this question. This would be one of those things like stains that we'd be using, differential stains, like Gram stains, like acid fast, like endospore stains. All of those things, they are differential stains. That's down at number 16, but they allow us to differentiate bacteria from one another. Some of that's based on their structures, like whether, what kind of a cell envelope they have. Are they gram positive? Are they gram negative? So that's determined based on their cell structure. That's determined by their cell envelope. Anyways, so that's one way. We can use PCR, which we'll get into detail in later chapters, but PCR is polymerase chain reaction. And that is a tool that is used. And I know people have heard of it because of COVID. Everybody knows that, you do a PCR test for COVID, that's the next step up from the rapid test. But that's basically just using the DNA from something and multiplying it a lot and then determining, is this DNA that we're multiplying, is it E. coli or is it not E. coli? Is it COVID? Is it not COVID? And that sort of stuff can be analyzed during the whole process by the machine itself. So it's a pretty useful tool, and it only takes a few hours in order to create billions of copies of DNA from just maybe a couple that we started off with. Super cool. So we can identify a bacteria that way. Let's just say about it. Physiological reactions in certain kinds of media, like the differential media or the selective media, whether or not they can grow on these things, whether or not they're able to survive or cause a color change, all that stuff is all information that we can use to determine different microorganisms from one another. All right, we have the observations of microbes and the concept of the cell. So Robert, I think it's Hook, I don't know how to pronounce it. Hook or Hook is the guy that came up with the concept of a cell being the unit of life. We have the guy that came up with the microscope to actually observe cells. That is Van Leeuwenhoek. So those are the guys that are involved in that. Magnification, refraction, resolution, contrast whenever we're using the microscope. Magnification, obviously, this is making things bigger, multiplying their size visually through the use of lenses, typically. We have refraction, that is essentially light scatter and how much light may be scattered whenever it hits a certain substance. When light moves from glass into the air, that difference in the media between the glass and the air causes the light rays to scatter because of that change in the media. So that causes a big change of refraction. Whereas if we add in that oil that has a pretty similar aspect to it that the glass does, it prevents that light scatter and allows for a clearer image. So that has how refraction works. We have resolution, which is essentially the clarity of the image, how clear or high def that image might look. And then we have contrast, which is literally just the ability to distinguish one aspect of something from another. So whether that's the cell itself from the background or certain parts of the cell from within the cell. That's microscope. So the major parts of a microscope, we went over those. I mean, when people come and watch this later, they won't be just in lab with a microscope lab day, unfortunately. But we just went over this today in microscope lab. The major parts of the microscope, you lift the microscope by the arm, you put your hand underneath the base. The slide sits on the stage, and you can move the stage around with the stage knobs. We have light shining in from the light source through the condenser that then focuses the light onto the specimen on the slide. And then you can control that using the diaphragm, how much light goes through that condenser. Then we have the objective lenses that allow for the actual magnification and creation of the real image when we magnify that. And then we have the nose piece to move around the objective lenses themselves to rotate between them. We have the ocular lenses that you're actually looking through. That also provides typically more magnification. And we remember that when calculating the total magnification, it's going to be the ocular lens magnification times the actual objective lens. So you multiply them. So that's pretty good. Did I leave any parts out, do you think? I think that's all the main part. All the interesting ones anyways. All right, we did just talk, I just mentioned briefly through that, the real and virtual images. Real is made by the objective lens, virtual is made by the ocular lenses. And we did talk about oil with resolution and preventing light scatter. The highest resolution of microscope is going to be the electron microscope because it is literally spraying things with electrons, and they're so tiny of little particles, you get all this detail. So you get the absolute most detail from the electron microscope. The two types of electron microscopes that are available is the transmission microscope and the scanning electron microscope. Transmission allows you to look like within cells and things like that, structures within cells. Scanning, we coat something with metal and then spray the electrons on it, and that gives you the image of the surface of something. Like when you see this really close-up picture, like spider legs or something. Heat fixing allows us to kill the bacteria that we put onto the slide, as well as attach it or adhere it better to the slide. And then it also allows for maintenance of the structures of the cell in the sample. So yeah, and then a differential stain, that is like the gram stain or the acid fast or whatever, but it's a stain that allows you to differentiate between different bacteria, whether they're positive or negative. If you do a simple stain, all bacteria is going to stain blue with methylene blue, right? But if we have a differential stain, now we can tell the difference between bacteria for whatever thing we're staining for. So I'll just differentiate. I like this one better. This is a better list. Okay. Chemistry. I didn't put too many questions about the chemistry on this one because A, most people have already taken chemistry and recently, and B, we incorporate it so much within a lot of these other chapters that the important aspects of chemistry just get reiterated anyway. So I didn't go over too much of the details here, other than probably the biggest take home from chemistry is pH and understanding how pH works and what it is. So I would say definitely understand about that. But covalent bonds, let's start from the top. Covalent bond is whenever electrons are shared between two bonded atoms. They're sharing those electrons back and forth. Ionic bonds, we have transfer of an electron from one atom to the other. And then whenever we put it into a solution like water, which is a polar molecule, it allows those ions to differentiate. So we have sodium and chlorine ions that can dissociate and interact with water, polar water molecules, because they're being ionized within that solution. So that's how ionic bonds work. We've moved like an electron from sodium over to chlorine because chlorine wanted its little orbits full, and the sodium said, I've got an extra one. And unfortunately, it leaves them with charges, and that means they interact differently with their environment. Now we have the hydrogen bonds. Hydrogen bonds are not bonds really technically. There's no sharing or exchange of electrons in any way. Essentially, some molecules like oxygen are more electronegative, and that just means they're going to hog electrons. And oxygen will hog the electron to it, so that gives it a negative charge because electrons have a negative charge. And that pulls it away from a bonded hydrogen, like what we see with water. So because we're pulling the electron away from the water is we're going to pull the electron away from the hydrogen in the water molecule. the hydrogen now has a positive charge to it, and the oxygen has that negative charge. We say hogging because they're hogging it. They're not completely transferring over. There's still a covalent bond going on there, but that electron spends more time with the electronegative oxygen. So that's actually what gives water its polarity. As oxygen steals away the electrons from the two hydrogen atoms, oxygen will have its negative charge and the hydrogens will have their positive charge. And so anyways, so your hydrogens now have positive charge for, they got their electrons hogged away. So they have a positive charge, allows them to interact with other negatively charged electronegative atoms out there, just due to that association of positive and negative wanting to be near one another. There's not any exchange across when we have interactions like that. So these are weak bonds. They are not like covalent bonds or ionic bonds are. So sorry, that's my phone. Okay. Crap, I don't care about. All right. So good. So that's how hydrogen bonds work. And those are actually the bonds that hold like our DNA, our double helix together. Those A binding with the T and the G and the C, which again, we'll get into that more. Can't you, be so excited about that in chapter 9? But the A's and the T's associate with one another. That bonding that they're creating are hydrogen bonds. And the whole double helix of one side of the ladder and the other is held together only by hydrogen bonds. And that's pretty impressive. So hydrogen bonds incredibly important in biology. They also give proteins their structure, the groups that are hanging off of an amino acid group that make it, instead of phenylalanine or alanine or tryptophan or whatever, those groups that make them what they are have certain charges or no charge. And then how they interact with one another determines how they're going to fold up and their overall structure. And the structure of the protein is typically what is going to dictate its function. So that's important, hydrogen bonds, and we will definitely come back to that. There's more to that on number 7, but we'll get to that. Right, so number 2, we have hydrophilic, hydrophobic, and amphipathic. Anytime you guys hear amphi, I want you guys to think like amphibians, right? They can be on the land or in the water. That's 2 different things they can do. So it's both, right? We're both hydrophilic and hydrophobic. How? We have a charged head on like a phospholipid that is a negative charge to it because it's got all these oxygens all around it. Again, remember they're electronegative, they're going to hog electrons. And then we have these fatty acid tails that are coming off that are non-polar. Those non-polar lipids are not going to associate well with water, just like oil doesn't associate with water. None of fats really do. But that charged head can, and so that gives phospholipids that make up our membranes an amphipathic character. They have hydrophilic, water-loving, and hydrophobic, water-fearing aspects to them. So that's what hydrophobic and hydrophilic are. Tends to be non-polar molecules are hydrophobic, polar molecules are hydrophilic. So that's number 3. That's the phospholipid that we were just talking about. And basically the charged heads will associate next to one another. And there's another layer of them down beneath them next to one another. And their little hydrophobic tails are stuck up towards one another in a way that allows them to protect that hydrophobic inside of those negatively charged heads. And that's what makes a lipid bilayer. That's what makes it do what it does. That's what makes it selectively permeable and allowing only certain things in and out of the cell so that we can contain everything within our cells. It's pretty neat. And that's all thanks to that amphipathic nature of a phospholipid. So pH is great and it confuses everybody. So pH is a measure of hydrogen ion concentration or hydronium concentration, but I don't want to get into that. So hydrogen ion concentrations. That's really all it is. And it's a logarithmic equation that gives us that value for the pH. So essentially, that's a base 10. So all of these numbers are base 10. So if I say you have a pH of 7 and a pH of 6, those are different from each other by multiple of 10. So if we're talking about hydrogen concentration there. The actual numbers, of course, is just one value in the number, but the actual difference in hydrogen concentrations, we're talking about a factor of 10 there. That's because this is all measuring as 10 to the negative, whatever your pH is. So 10 to the negative 7 is a pH of 7. That is whenever we have an equal number of hydrogen ions and hydroxyl or OH negative groups. And when they are equal, that's water. So that's what everything is based off of. So anything less than 7, a pH less than 7 is going to be acidic, and pH greater than 7 is basic. But how does that work when we're talking about this in terms of the concentration of hydrogen ions? So acids, we know those are going to have a lot of hydrogen ions. So if we have more hydrogen ions, why is the number less? Because it is 10 to the negative blah, blah, blah. So 10 to the negative 2 means if you start with like a decimal point and you have a negative 2 and you're making it 2 decimal points smaller in value. Okay, so if I have 10 to the negative 2 versus 10 to the negative 14, which is more, just in general as a number, 10 to the negative 2 is more than 10 to the negative 14. Moving it over and making it less by 14 spaces, whew, that's a lot less. That is the measure of the hydrogen ion concentration that we are talking about there with the pH. So 10 to the negative 2 means a pH of 2, and that means 2 has more hydrogen ions than a pH of 14 does, which is 10 to the negative 14. Okay. When we have bases, which are those ones that are higher value, pH value than 7, so getting into 8, 9, 10, 11, 12, 13, 14, the more basic that we're getting, the less hydrogen ions that we have, but the more also hydroxyl ions we have. And that's the OH negative. And why are we talking about those? That's because all of this has to do with water. So if we're talking about hydrogen ions, what if you take a hydrogen off of water? What do you have left? An OH. That's a hydroxyl group. So that's why we are comparing those when we talk about pH, when we're talking about acids and bases. Remember that another word for base is alkaline. Okay, so that's pretty good on the pH stuff. I don't know, I feel like, but how do you guys feel about the pH stuff? Is it okay? If you guys have time to like look over this and study it and quiz yourself and see if you can understand this stuff and you're still not getting it and you want me to find different ways to explain it, just let me know. And I'll see what I can do for you. You can always email me about it anytime. And I'll try to see what I can figure out. Because I know the pH, that's a really difficult concept. And so it's okay if we're having trouble with that. All right. And really, if your only take home from this course, of course, I'm going to quiz you more in the course about this stuff, but if your only take home from the course is that you know that, acids are less than 7 and bases are greater than 7, then hey, it's something. It's something. All right, organic molecules, most of us should know that by now. This is carbon hydrogen based molecules. Anything that isn't carbon hydrogen based, we call those inorganic molecules. That simple. 4 major biological macromolecules. We're talking about the proteins that we already mentioned. DNA is a nucleic acid, so the nucleic acids, we mentioned those. We've already talked about fatty acid tails and how they don't like water, they're hydrophobic. Those are the lipids. And then we have the carbohydrates, which are sugars. Okay, so, right, so the monomers for these guys, for the sugars, the monomers are going to be monosaccharides, which are just single sugar units, and they can be bound up to one another over and over again to make things like cellulose, to make things like starch. For the lipids, they have like fatty acids essentially, and they have their hydrocarbon tails. For the nucleic acids, we have the nucleotides. We'll talk about those more in chapter 9, but a nucleotide is the structural functional unit for both DNA and RNA. And DNA, well, both of them, a nucleotide means that we have some sort of nitrogenous base. That's what determines if it's A, T, C, or G. We have nitrogenous base, then we have a pentose sugar, which is just a 5-carbon sugar, and then we have a phosphate group. And that's what all nucleotides have. For DNA, the sugar is deoxyribose as opposed to ribose for RNA. It means an oxygen is removed. And then for DNA, you also have A, T, C, G, whereas RNA has A, U, C, G. But okay, so that's a difference there. ATP, that's an important one, adenosine triphosphate. That is the energy molecule for cells. That's as exciting as that gets. Sorry, but we'll get into that in chapter 8 when we talk about metabolism. But that is a nucleotide type molecule. Adenosine is like the adenine, which is a nitrogenous base that DNA had. It has a 5-carbon sugar. For ATP tends to be ribose, which RNA is ribose. RNA is our messenger molecule from the DNA blueprint. And then we have the phosphate group, except in ATP, we have 3 of them. And phosphates have a phosphorus with a bunch of oxygens around them. Those oxygens have that negative charge, remember, because they're, going to hog all the electrons. They're greedy. So that will have all these 3 phosphate groups right next to each other, all negatively charged, just pushing against one another. They don't want to be near each other because they're all negative, just like south poles of a magnet or something like that. So that's how the energy is stored, because if we can break that bond and release that tension, the cell can actually use the energy released from that association for other functions. So that's how that would happen. That's in chapter 8. So that is related to the nucleic acids. Proteins. Okay, so we did talk about, we've already mentioned that the hydrogen bond and how important they are for the shape of the protein. Protein monomers are amino acids. Amino acids can be all these different kinds of amino acids, and they can have polar charge or they can be nonpolar and interact with one another the way that those molecules would in chemistry in general, right? So that helps determine their shape. The amino acid sequence of a protein constitutes the primary structure of the protein. So it's just an amino acid sequence. How those amino acids interact with one another allows them to make structures like alpha helices, exactly what it sounds like, just a helix, or beta sheets, which is a beta pleated sheet is just like a smooth back and forth area within the structure. That's the secondary level of the molecule. The tertiary level has to do with now how farther away polar or non-polar molecules might now be interacting with one another based on having made the alpha helices and the beta sheets. So more associations of those groups, polar or non-polar, as well as interactions of sulfurs across from one another. They can actually create disulfide bonds between amino acids with covalent bonds at that point. And then if we need to have those tertiary structures, which is that third level, come together to work together as a team to create a functional protein, that constitutes that functional protein is the quaternary structure. So that's the 4 different levels of structure for the proteins. Yes, so that's chapter 2. So I think that's everything on there. Oh gosh, okay. I didn't realize that this was so long. Okay. Chapter 4, this is the last one, so we just have to make it through. I'll try to kind of, I don't want to say rush, but I'll give you the rundowns instead of like all the little details. So differences between eukaryotes and prokaryotes, we know about the membrane-bound organelles, the big ones, right? For eukaryotes and prokaryotes, membrane-bound organelles are in the eukaryotes. Prokaryotes don't have that. Prokaryotes, they don't have a nuclear membrane around their nucleus either. So they basically just got a cytoplasmic membrane that just contains everything floating around in there. So that's the idea with those. Prokaryotes also tend to have a circular chromosome, whereas eukaryotes have linear chromosomes. An organism is considered single-celled if that individual cell can come off of the biofilm or whatever it's associated with and continue to divide and live, and it doesn't need the rest of the cells. If you take one of our cells off of our body, it's going to die. It needs the rest of the cells there to support it. So we can't be single celled. That's how you determine what a single celled organism is. The 3 general shapes of bacteria. The big 2 that you guys need to know of, we learned about today, the cocci, which are the round ones, and the bacilli, which are the rods. We also have the curved ones, and we'll just leave it at curved. They can have spiral shape or like a comma shape or whatever, but curved is the only thing you really need to know about it. Common arrangements, we have staphylo for irregular bunching. We have strepto, which I think of strepto, those are the chains of the cells, end to end. We have diplo, which is pairs. We have tetrad, which is 4 together. We have the cubes, which is known as sarcina. And we have palisades for the bacilli, which is just when they're attached at certain points, but are stacked up next to each other, sort of like a fence. That's a palisade. Flagella, pili, and fimbriae. Flagella are for movement. Pili and fimbriae are for attachment. And pili is also used for moving DNA from one cell to another through the sex pili. It's called conjugation. Okay, arrangements of the flagella. Flagellar arrangements can be polar, or they can be all over. Polar means at one end or the other or both ends. The polar arrangements include monotrichous. That term means one flagellum at one end. Lophotrichous, which means a tuft of flagella at one end. Or amphitrichous, which means flagella at each end. Then the word for having flagella all over is peritrichous. E. coli, for example, is lophotrichous. Chemotaxis and phototaxis, the cells can respond essentially to these as signals. If you were an organism that needed to, undergo photosynthesis, then you might react very excitedly to sun also. But anything that cell might be interested in wanting to use as food or something like that, they can react to via chemotaxis. It's just chemical signals that draw it in a direction. They can also move away from things due to negative signaling, if it's like a toxin or something like that. So cool. So we understand about that movement. Runs and tumbles are movements that the cell will do using the flagella. Remember, bacterial cells, they rotate their flagella. They don't move them in a whip-like motion like eukaryotes do. So prokaryotes are going to rotate their flagella so they can like go straight while they're rotating it one way, and then suddenly move the flagella the other way. And then that allows them to create a sudden change in their direction. And then they use that and allow them to move towards or away from certain things. So runs are moving straight, and a tumble is that change in direction. Bacteria do not reproduce sexually. They undergo, binary fission, which is like you start with one cell and then you divide it into 2 cells, and they're exact copies of that one another. But we do talk about the sex pilus and conjugation, and that is whenever we have little tunnels that are made between bacterial cells of the same species, and we can move DNA like useful DNA traits like antibiotic resistance can even get moved between cells that way. So this just allows us to move DNA pieces. The glycocalyx is a sugary, sometimes protein is associated with it, coating that is very sticky. We have the slime layer that is loosely associated, or the capsule that is very tightly associated. So that's the 2 features that fall there. The capsule allows for evasion of phagocytosis. That means that our white cells that are trying to attack and eat up these bad bacterial invaders can't eat them as easily if they have a capsule that's sticky coating. So that is a useful trait for them to have if they are a pathogen. So that's the capsule. So the biofilms, what's the medical significance? Biofilms can cause coatings on things like internally surgically implanted things like pacemakers or other structures like that. if bacteria are able to contaminate those surfaces or in the crevices of those surfaces. We can also have that that can lead to endocarditis if it's associated with the heart-related piece of structure, medical equipment. And then we have plaque, like dental plaque, that's caused by biofilms. Biofilms are, again, bacteria that are living very, very closely associated with one another, usually stuck to one another or stuck to a surface, and they multiply like that. And then they also can communicate with one another based on signals, chemical signals that they can send that tells them how well the biofilm and the organisms in it are doing. So plaque and growth on medical implanted devices. The cell envelope of bacteria and the gram-positive and gram-negative. Cell envelope is the outer coating of the cell. The bacterial cell, we have an internal, all of them have a cytoplasmic membrane. That's just the membrane that, surrounds everything in the cell. All cells have to have that, and all living things have that. So that was part of the requirement for the cell theory. So we know they have the cytoplasmic membrane. Most bacteria also have a cell wall of some kind. If it is a thick layer, that is gram-positive. If it is a thin layer of cell wall, then it's gram-negative. Also, gram-negatives will have another membrane on the outside that allows them to, well, it's just a membrane on the outside. And some of the stuff that is stuck into their outer membrane is called a lipopolysaccharide. I mean, remember, lipo, fatty, polysaccharides can be sugar molecules, right? And that lipopolysaccharide is what we call an endotoxin. It's only associated with gram-negative bacteria, but it can cause shock and fever in humans who are infected with gram-negative bacteria. What else? The peptidoglycan itself is a repeating sugar subunits of it's N-acetylmuramic acid and N-acetyl glutamate or glutamic acid. NAM and NAG repeating and linked together. So that's sugars, right? So that's like glyco, like glucose, sort of sugars. And then peptido part is literally peptide linkage. So amino acids that connect those long, chains of sugar. So that creates a scaffolding that's very, very thick for gram-positives. They also have lipoteichoic and teichoic acid. These are just features they have that help them whenever they're dividing to make new cells, to help them keep, their structure of their thick cell wall. So they need a little help with that. They don't have the outer membrane. Gram-negatives do, but gram-positives do have lipoteichoic and teichoic acid. So it's the cell walls and how we differentiate those guys. Cell walls of mycobacteria. Mycobacteria have mycolic acid in their cell walls. So they're considered gram-positive, but they have this wax called mycolic acid. It's very waxy and protects them from certain harsh conditions, as well as cleaners and all of that. So they could survive that stuff using this wax, makes them resistant. They stain acid fast because of that. So these are the acid fast bacteria, mycobacteria. The mycoplasma are a different group. They don't have a cell wall at all. So they just don't have one. So they have a lot of different shapes of their cells, because a lot of times cell wall determines the cell shape. So they don't have that. So they're pleomorphic is the word for that. Pleomorphic means different cell shapes. Gram-negative bacteria have endotoxin. We talked about that. What is it? It's the lipopolysaccharide in their outer membrane, and it's important because of the fever and the shock. What makes gram stains work? So our body automatically sees that as foreign if it's going around. That's important. So what makes the gram stain work and what happens at each step? I am glad to explain that to you. So the gram stain works. We have crystal violet, the purple stain, and it's going to be added to gram-positive and gram-negative cells, and it gets stuck in the cell wall, just due to its size and everything like that. Then we add in iodine. Iodine is a mordant. So crystal violet is the primary stain. Iodine is added as a mordant to help stick everything in place. Then we rinse with alcohol. Alcohol breaks down the outer membrane of the gram-negative bacteria and allows for better rinsing out of any residual crystal violet. It won't stick, the crystal violet won't stick in the cell wall of a gram-negative, it's so thin on there. So that's one way we rinse out all of that crystal violet with alcohol, but it stays in with the gram-positives because they're so thick. We trapped the crystal violet in there and we stuck it in place with the iodine. It ain't coming out with no alcohol. So at that very point, now we have a differentiation already. Gram-positives are going to be purple still, and the gram-negatives now are going to be colorless because we removed it with the alcohol. Now we will counter stain with the safranin, which will make gram-negatives red. And I always think negative associated with red and purple positive, PP, purple positive. That's how I remember which color is what, and we'll learn more about that in the lab on Monday when you guys get back. That's the next thing we're doing. Selective permeability means allowing certain molecules to get into the cell and not others. So like water and other small molecules might be able to pass through, but larger ones can't get in and out without some kind of a help going on. Plasmids are DNA, extra bits of DNA. They are circles. They're much smaller than the bacterial chromosome though. And they can code for things like antibiotic resistance or the production of certain toxins and all sorts of stuff like that. That's how they're medically significant. We can also use them as tools for genetic engineering. With endospores, endospores are protective structures of some bacterial cells that allow them to survive harsh conditions. Endospores, basically what's going to happen is you have a vegetative cell. That's the normal metabolically active cell. Vegetative cell has some sort of harsh condition, usually lack of carbon in their food source, nutrition. So they decide to hunker down and wait till things get better. So they go through this process called sporulation. That's forming the endospore. The cell that is doing that is called the sporangium. So it forms its little endospore within itself and covers it with what's called a cortex layer that's just going to make it resistant. Now that cell has made the endospore that is resistant to so many things, you guys. Oh my gosh, like radiation, cleaners, and heat, even like certain kinds of like boiling, it could survive that. It's nuts. So these guys can survive pretty much like just anything you can almost imagine except for like an autoclave. So these guys are super resistant and stick around. They're so resistant that you can make powder out of those dried up little harsh endospores. We take powder of that and put it in an envelope and mail it to Congress. That's anthrax. So that's how harsh, like that's how much these guys can like resist things. Just put them, dry them out into a powder and they're good to go. They don't even have to be reconstituted. So right, so these guys, so that's endospores, and if they, get into, let's say they're in a spore form like anthrax, and you inhale it, let's say, you breathe it in, and now it's found its nice amounts of carbon, it's found its nice amounts of whatever other thing it needed that it was waiting for, it's just waiting for, and it will go through germination at that point and turn back into a vegetative cell again, normal cell now, it just pops out of its little coating. Only takes it 1.5 hours to do that. That's endospores. I already explained the steps of it. Archaea are like bacteria. They look like bacteria to us, but genetically they are more like eukaryotes than they are other bacteria. Archaea tend to be extremophiles. That's the type of habitat they would typically thrive in. And what do I mean by that? So super hot temperatures. I'm talking they flourish above boiling temperatures. I mean, it's nuts, right? Because these are guys in the thermal vents and stuff in the bottom. These are the methanogens, the ones that make methane in swamps and that sort of stuff. Can survive in freezing temperatures. If they can survive a high temperature, then they typically can also survive high saline or high salt content, which is a halophile. And they also can survive acid as well, typically, acidophile. So they are the extremophiles. The 4 groups of bacteria based on their cell envelope. Oh my gosh, I'll have to write it down. So let me go through these. Gracilicutes. They all end in acute. Gracilicutes, they're not cute though. Let's see, Mendoza-cutes. Tenericutes. And the firmicutes. Okay, so those are the 4. I break them down like this. I might write down all 4 of them like I did just now, because this is how I had to remember it. The firmicutes, I remember they're the ones with the firm cells. So these are all based on like their outer covering of these organisms, right? The firmicutes, these guys have a firm coating. So these are going to be your gram positives. They have the thicker cell wall, right? They have the firmer cell walls. Then we have the Mendoza-cutes. This is always my next one. These are the archaea. I think of archaea as being like archaic or old. And I had this boyfriend in middle school whose name was Joey Mendoza. And so I think Mendoza-cute as being like back, ancient, ancient boyfriend. That's going to be Mendoza-cutes. Okay, so we've got gram positives and we got archaea with the firmicutes and the Mendoza-cutes. The next one that I always knock out is going to be the gracilicutes. These are just your gram negatives. So it's the G1. So the other G is gram negative. And so the last one that we have are the tenericutes. And these guys have no cell wall at all. And these are like the mycoplasma. Okay, that's it. We did it, we survived it. It was about an hour and we didn't die. So let me go ahead and stop this recording so people don't have to listen to me ramble anymore.